Great. Thank you so much. Next up, we'll hear from Bjartmar, CMO of Augustine Therapeutics. And he will talk to us about targeting cytosolic HDAC inhibition for the treatment of CMT and beyond, from basic research to initial clinical studies.
Thank you. And thank you to Laura and the CMT Research Foundation for this opportunity to speak about, I think we should have another slide set. For this opportunity to talk about our HDAC, that's better. Our HDAC6 program for CMT. So this is basically be the story from the laboratory into the clinic. So we are Augustine Therapeutics, and we are based in Belgium, in Leuven, although we have colleagues from many geographies, including the Boston region. Myself, I'm Swedish. And so what we have is a discovery platform with a next generation HDAC6 inhibitor. And I should already now point out and emphasize that HDAC6 works. It works in the cytoplasm, right? So it's non-epigenetic, and I'm going to sort of repeat that a couple of times here because it's important. Our lead program that's a long name here. I call it 216 for simplicity, and the indication is CMT. And I will show what we believe is very promising data in animal models of CMT. And we are, as mentioned, currently in phase 1. The phase 1 study started earlier this year, and we expect to have the full picture of the data later this year or early next year. There's also in this platform additional candidates coming up with various potential. There's a huge potential here with various indications, both neurological, neurodegenerative, but also cardiometabolic. And I should also mention here that, and this is as a small biotech company, this is important. We are backed by very dedicated and strong investors, both strategic and venture, which, and that's the point here, enable us to move this program well beyond phase 1, should the data allow us to do so. Again, that's important for a biotech company. So HDAC6, it's an interesting compound. It's interesting molecule, interesting enzyme, and it is unlike other HDACs. It's a cytoplasmic regulator that's involved in a number of cellular processes, and some of them are listed here. It's microtubule transport or axonal transport, enabling the transport of mitochondria and other organelles, cargo throughout these long axons. It's also involved in oxidative stress, neurite outgrowth, regeneration, protein clearance in the case of CMT, PMP22 misfolded, for example, and also immunity and inflammation. So there's a lot of potential effects here. And again, the effects are not mediated through the nucleus. It's not epigenetic. It's in the cytoplasm. It's therefore also a druggable target. So here is this illustrates our journey. And there's a photo here of, this is Ludo van der Bosch. Ludo is over there. And that's really where this story started some 15 years ago, when Ludo and his team identified HDAC6 inhibition as a promising tool or promising approach for neurodegenerative conditions, including CMT. And it was initially CMT2 and ALS, FTD. And we have subsequently also moved this into not only CMT2, but also CMT1. So in 2021, we identified the 216 as our lead compound. And we generated the first sort of proof of concept in the CMT1A model. And we are, as mentioned now, currently in phase 1. And this is our building, this is our lab and there we go. So we talk about unmet needs. Of course, for CMT and patients living with CMT, there's a huge unmet need. That's obvious, and that's why we're here. But when it comes to HDAC6 inhibitors, specifically, there's also an unmet need. So we need compounds, inhibitors that have a better PK profile, reduced toxicity, and improved efficacy compared to the earlier generation HDAC6 inhibitors. So again, our platform has this next generation highly selective HDAC6 inhibitor that we believe will overcome many of the shortcomings of the earlier generation HDAC6 inhibitors, including target effects and our chemotype has again a selective inhibition, and as far as we know, no off-target effects. It's been very well tolerated in non-GLP and GLP tox studies, which then has justified us to move into the clinic where we are now with the lead program 216. But again, and for CMT. And also, as mentioned there, we have a pipeline of compounds in the background coming up with potential neurodegenerative and cardiometabolic indications. So the mechanism of action for 216 is partly based on the tubulin hypothesis, which is based on that in pathological conditions, in stress conditions, HDAC6 will drive the deacetylation of molecules and deacetylation of alpha-tubulin. And that leads to an unstable microtubule, decreased axonal transport, and reduced viability of the axon and of the mitochondria, which drives the pathology in CMT. So by inhibiting the enzyme with inhibitor, one can restore and partly rescue the phenotype of the microtubule, and then and restored axonal transport and axonal viability. And we know that axonal loss and axonal dysfunction, it is feature of both, not only CMT2, but also CMT1. And then there's this picture here, and if I push this one, it should be, maybe you can see that it starts moving. This is a so-called mitotracker, which can quantify the movement of mitochondria along axons. So these are cultured motor neurons. And in this assay, it's also demonstrated by Ludo here that with HDAC6 inhibition, you could actually increase the motility of mitochondria along the axons. So here is our non-clinical data, and this is the CMT1A model. This is the C3 model that was mentioned earlier here today. And this is a symptomatic treatment model. In other words, the mice at 8 weeks are clearly symptomatic. And then the treatment begins for 4 weeks until 12 weeks. And in this study, there were 4 different doses, and there were daily dosing again for 4 weeks. And as you can see here, both for CMAP, which is here, and for conduction velocity, there's a clear dose-dependent partial rescue of the phenotype. And we have very similar data for grip strength, and I don't show that here, but it looks very similar, dose-dependent rescue in improvement of grip strength. And then if you look at the corresponding histology compared to the wild type, you can see that there is regeneration and remyelination compared to the untreated animals. So this is, we believe, extremely interesting. And then if we look at it's kind of a biomarker, right? As target engagement, tubulin acetylation that you can measure here. There is, compared to the untreated and to the wild type, there's again a dose-dependent increase in tubulin acetylation that we can measure. And then it seemed to, and that's the case for all these graphs, it seemed to plateau at the highest, at the two highest doses. And then biomarkers were mentioned here earlier, neurofilament. We looked at neurofilament. You can see in the same case here in the untreated, you see very high levels of neurofilaments, clearly elevated, and there's a dose-dependent reduction in neurofilament in this model. So that's the animal data I'm going to show here, and I know that other labs have very similar data also in the CMT2 model, and we think that's also, of course, very interesting. So where we are now, we are in phase 1. This is a very typical first-in-man study design, where the objective is, of course, primarily safety, efficacy, and then to find the right dose. There's a single dose part of this, and then a multiple ascending dose part where the subjects are treated for 14 days. But in addition to the conventional endpoints and readouts, we also have, we can see that here it's an exploratory readout. It's PD, there it is. And that's blood sampling of acetylated tubulin, so basically tubulin acetylation, which is serves as a biomarker or a marker of target engagement in humans, and see if we can sort of translate and reproduce what we saw in the animals. And that will, of course, guide us then when it comes to moving this program into later phase studies. And then just a few words to wrap this up about the substrates. So again, as mentioned here, there's been focus on, I talked mostly about the acetylation of tubulin as the primary translational PD marker, which we are looking at that with interest in the phase 1 study. But then I think tubulin acetylation and axonal transport doesn't explain all the effects we see in these animal models, right? Especially CMT1A, such as regeneration and remyelination. So there are probably other mechanisms involved here and other substrates, and HSP90 is of interest. Cortactin is of interest for the remyelination and regeneration we see. And there's also data that in this model there's also suppression of PMP22 levels. So there are clearly other things going on here, and that's something that we are looking into now as we speak, and try to learn as much as possible about the mechanisms behind the clinical effects or the effects we see in the animal models. So that's where we are now, and I just want to close out with these are some of my colleagues, and Gerard is here in the room, and again mention that we are fortunate to be backed by dedicated, strong investors that again enable us to move this program forward. So to wrap this up, what we have is a unique chemistry platform for a next generation, very selective HDAC6 inhibitor that hopefully can overcome the shortcomings of all the earlier generation inhibitors. We are now in the clinical stage with 216, and of course, the indication is CMT. We have additional pipeline compounds coming up, and we are fortunate to have leading investors backing us. So I stop there and happy to take questions.
We're going to start here because you were the last person last. Thanks. Hello, great talk. My name is Mary McMahon from Revere Therapeutics. So HDAC6 has also been implicated in stress granule formation. I'm wondering, have you guys looked at that in any of your models? And second question, do you see, have you looked at the alpha-tubulin acetylation in patients? Is it altered?
So no, we are currently in healthy volunteers. That's absolutely something that we are going to look into.
Great, thanks very much.
Yeah.
Hi, Bruce Carter from Vanderbilt. My question is, if I remember, there was some data that the HDAC inhibitors were helping clear PMP22 through an autophagy mechanism. Are you aware of that data, and have you looked at autophagy in your effects of the compound?
That's not something we have looked into as far as I know. No.
Okay. Thank you so much for your talk.
Thank you.